Okay, so that's a lot to live up to. So our guest speaker tonight, is anybody here not familiar with the name Chad Cotter? The front row, the speaker himself is not familiar. And you have to remind me the title of your book because it's been out for a while. The Organic Mushroom Farming, and so Trad comes from South Carolina originally, and has a wealth of background with cultivation and other mushroom related activities. But I don't know if I should mention the Blue Portal. Okay, and so the Blue Portal is I guess the clinical service, clinical retreats that are offered. I know in Jamaica and Costa Rica as well. And so if you're, hopefully you'll talk some about those as well. But has been a long-standing member, faculty member of the Telluride Mushroom Festival. And I was first inoculated with his wisdom years ago. And he's got a lot to say. So help me welcome Chad Cotter. What do we want to shift this off? So where did Joe go? I just don't want that showing.
Do you know how?
Because it's on Zoom. Yeah, no, I was looking and this wasn't working. Yeah. Is that, yeah.
The speaker.
Fast forward, just go right.
That's right, arrow, yeah.
Oh, that's pretty dumb, dude. I'm sorry. Maybe wear sunglasses.
No, my sunglasses. That's why I brought my sunglasses. Use my prescription glasses. Can everybody hear me okay? Good. All right. Thank you for coming tonight. It's an honor to be here. We planned this, what, a year ago? I think it's been almost a year. Come out, really busy and travel a lot. I just want to ask you, we're going to be talking about mushrooms, molds, and mycorrhizae, and all the amazing things that fungi do. How many of you are growing mushrooms right now legally? Well, I'm in Colorado. See, if I was in South Carolina, everybody's hand would go down. But I'm going to ask you, do you want I've got 2 PowerPoints pulled up. Do you want a fun and entertaining deep dive, or do you want an academia chewy one?
Titles. Yeah, you get those titles?
This one, they're both the same. Fun. Yeah, I've done enough mushrooms in my life where I'm psychic. So we're going to do the front one. I didn't have 2 planned, by the way. All right. So you got some morels up here too, by the way. Greg found some morels here, some beautiful morels. This is how big morels get in my area. These are, ash morels. They get pretty big. And I found 168 around one tree. And you have to wear camouflage if you're going to find them. But I'm going to start really easy. We'll make some jokes and we'll jump right into the thing. So it's going to be a funny lecture, I hope. But after my divorce, I'm going to tell you that I was on some dating apps, and we're going to start like that. So we're going to start easy. I told you. I was on some dating apps, and I really wanted to meet someone who was into mushrooms. So I was like, hey, do you like morels? I thought, hey, that's a good pickup line, right? Does that work for anybody? So I'm like, hey, do you like morels? So I send, and it zips off and it's gone, right? You can't return that text. And then I looked down and it auto-corrected it. And instead of saying, do you like morels, it said, do you like motels? And I still didn't catch it. And then here comes the bubbles. Excuse me? And I'm like, have you ever been motel hunting? I have a couple motel spots and we can hit that are really close to each other. Anyway, let's get to the lecture. I just want to say that going back in time, this is a journey. This is a storytelling event where this planet has been through a lot. We know that, and it's going to go through a lot. It's going through a lot right now. And I'm very interested in life on this planet, how it began, how it is right now, and what's going to happen in the future. It had all the right chemistry to produce something that was just amazing. Molecules, DNA, amino acids, in DNA, and then we have these organisms that just, they just, how did they appear? It's like, how did they evolve? Prehistoric fish, and life evolved in the ocean. And then we had, land mammals, and reptiles. We have this evolution of the ocean to land and plants. This is millions of years ago. So what happened? I think we all know what happened. Or it's speculated, right, that a very large chunk of rock unpredictably hit the planet, came in at millions of miles an hour and slammed into the earth. Will this happen again? Right? Will it change the course of history? But you know what was there? This planet didn't go extinct entirely. Philosophers have said there has to be destruction to have construction, right? So we lost a lot of species on this planet, and we continue to lose the species on this planet.
Sounds like you're in.
Oh, I got you. Yes, you're looking at dead dinosaurs if you're on Zoom. But something was there, like in the cracks, in the crevices of life. Life did survive. Little molds, little microscopic algae, little plants, little tiny voles, little tiny rodents, in the cracks, somehow made it with no sunlight. And so life finds a way. Life finds a way. And this is amazing, right? And we're going to talk more about these plants. But more so is how these bacteria, archaea, which are the most basic. And if we look at, I don't have a laser pointer, but all we see over here are fungi and animals and plants. Those are the majority of the visible animals and plant life and life that we actually see. The rest is invisible, right? Or without instruments, without microscopes, but they're there. And so we have protists, we've got slime molds that are covering. They're like little janitors. They crawl around. They're not fungi. They do produce spores. They're like little herds of cattle. They crawl around eating bacteria and cleaning the woods. And they're very smart. They're very chemically intelligent. You can look up these. They're finding the way through mazes, and they can map the subway system of Tokyo, and redo our DOT system. Just phenomenal. Absolutely amazing. But something else happened, was the merging of life, which is composite organisms, which are bacteria and fungi and algae together as lichens. Lichens right now currently cover as much as 60% of this planet. I know you walk outside, you're like, I don't see lichens here in Denver. I don't see them in my yard. But you've got to go to the surface area of the mountains, the whole entire planet. Lichens and mosses. Photosynthetic, right? And these are beautiful. Carry a little, I got a hand lens in my thing. I keep it all the time. I walk around. These are aspen seeds. They've got little cup fungi, just like a morel, right? And then we have mushrooms emerge, right? These beautiful, these are oyster mushrooms emerging. I've been growing mushrooms, for 32 years, in my room over the garage. When they asked me at 20 years old, what are you growing upstairs? And I said, I'm not going to lie to you, portobellos. And my mom was like, they don't look like portobellos. Yeah, why are they bruised and blue? But then other organisms, millions of years ago, figured this out. This isn't a fossil record. They got ants that are cultivating fungi. These are really cool, right? Really awesome. And I do have property in Jamaica and Costa Rica. Costa Rica, I see the leafcutter ants all the time. Ants are probably one of the most populous group of organisms on the planet, right? And they're very efficient. They're very organized. And insects are the largest kingdom on the planet. fungi are number 2, right? In these leaves, they select these leaves for a reason. There are fungi in the leaves that they can detect already, right? They take these leaves back, they chew them up. Where is it? I don't know where my one slide went. Anyway, they make fungal gardens with these leaves, and they chew the leaves up. They even, what goes in must come out. So their excrement goes onto the little fungal garden and also acts as a trigger to detoxify the fungal garden. And in return, the ants receive food and protein and a natural antibiotic that protects them from infections. And when the queen leaves, she has a little pouch right here. Everybody touch that little part in your neck. Y'all feel that little part in your neck right there? Little depression. You're the queen. They carve out a perfectly little piece of mycelium and they put it, and it fits right into the ferabucal pouch. And then it fits in there and they leave and they start a whole nother colony. And with that one little tiny pellet of mycelium, like an ember, it's amazing. Then they start the whole process over. So they know how to make spawn, right? They don't have a lab. So ants are doing this. They're also good, very well cultivators of Lepiota, the group Lepiota. I've taken Lepiota mushrooms and ripped off the caps and just stood over ant piles and just ripped off the caps. And I'd watch the entire colony of ants stop, and the guard would come out and get up on his back legs and start flailing around. And the ants would stop gathering twigs and seeds, and they would start dragging the mushroom caps down underground. You have to observe, you have to play around, you know, with life. You've got host jumpers, you've got rusts. You've got, anybody know what this is? This is cedar apple rust. Looks like a little kush ball, right? Remember the old kush balls? Some of you don't. They're really jelly-like. They come out of the pods on the cedar, and they also infect apple trees. So these are host jumpers, and rusts will jump from plant to plant, and they cannot go from cedar to cedar. They have to go to cedar to apple and back and forth. And there are some rusts that go multiple species of plants, 5, 7, to get back to where they need to go, right? Very complex. I'm very interested in rust for biocontrol of native or invasive, or excuse me, invasive plants, you know, especially things like kudzu and poison ivy. Do you have kudzu here? I brought you some. I just planted it out back. Lecture's over. And then there's corn smut. Anybody ever had corn smut? I love corn smut, right? You could call it a mycoparasite. It's really just it infects corn kernels. I grow it. I grow it intentionally. You can take a corn smut dried kernel, put it in distilled water and inject it down the silk channel of a species or a strain of corn that is susceptible to it, and you can grow your own corn smut. It's highly, way more beneficial than regular corn, highly more valuable. Corn farmers in the United States get 1/10 of a cent for every ear of corn, 1/10 of a cent, and these are worth $1 an ear, right? So you can, if you're not familiar with corn smut, you can search that. Just be careful about what pops up on your phone when you search smut. Make sure you put corn, or just scroll down until you see corn. Mushrooms create heat, carbon dioxide, and they sweat, right? They sweat and they swim through their own sweat like this. Kind of like, this is how I envision. I'm almost a mushroom. I'm really close to evolving. And you have to think like a mushroom. So mushrooms create heat, carbon dioxide, and they sweat. They swim through their own enzymes. They make enzymes. Those enzymes combust with oxygen. That's the oxygen, combust the molecules. That's why there's heat production, right? And they sweat water and they swim through that chemical fluid of enzymes and antibiotics. And they swim through there and break down their food source and absorb it right through their tissue, right through their cell walls. Kind of like an animal turned inside out, right? Mushrooms also, fungi also produce octanol, some more than others. Octanol is a mushroom alcohol. We produce octanol through the pores of our skin. It's a fruity smelling alcohol. And that is exactly how mosquitoes find you, right? That is the key to a future mosquito repellent is tamping down octanol coming out of your skin. That's why runners, when they run in sweat beads, I mean, that is octanol. It is you are attracting flying insects, right? And so what also is attracted to octanol are worms. Also all kinds of different insects, butterflies, bees, and termites, by the way. That's why termites really are more attracted to wet infected wood. Let's like wet wood. They're more attracted to the mycelium that's in the wet wood, and that's what attracts the termites. It's the octanol. So there's the future of termite traps, right? But when you're gardening, if you have mycelium in your garden, if you are growing King Staphyria and wood chips, or you have mushrooms fruiting out of your yard, that's a great thing because that's attracting other organisms that want to eat the fungus. They're going to make deposits. And that's going to make natural fertilizer for your plants. So you're not already growing mushrooms in your garden, you should start. Or invite the fungus to your garden. Bring in wood chips, bring in organic debris, plant some spawn, just mix it up in there, and they could be residential. Plants have been around a long time, right? I don't think they're going anywhere. They've figured it out. Millions of years. They figured out how to make relationships with different organisms and fungi, especially. When their roots start to invade and go down into the soil now, this is millions of years in the making. They are looking for fungal partners called Mycorrhizae. Right? These are symbiotic fungi that enter the cell walls, around the cell walls. They don't actually penetrate the cells. It's like taking your fingers and pushing on a balloon, but not popping it. It makes it increases the surface area of that cell with these little finger-like projections in or around. There's endo and ecto mycorrhizae. And they also can communicate from plant to plant, right? They can cross-stream different food sources, nitrogen and phosphate, to the plants. As much as 30% nitrogen is streamed through a mycorrhizal relationship and 60% more phosphate. And the plant's running out of phosphate, mined phosphate. So fungi naturally do this. They naturally regulate nitrogen and phosphate to the plants when they need it. But humans, we decided that we want to get granular fertilizer, dump it on the roots, because we know exactly what they want when they want it. Pretty wild, huh? There's no way to understand the cellular regulation of a plant and a fungus. It's like, it's dynamic. It's happening all the time, right? Sugars flow down from that tree or the plant. As much as 60% of that paycheck from photosynthesis gets sweat into the soil, and that is utilized by the microorganisms in the soil. So something is coming back, right? Trees give back to the soil. That's how important it is. It's a huge network, thousands of miles of mycelium in a square inch of soil. You have protection from pathogens here. You have nutrient absorption. You have water absorption. You have stability. You have adhesion of the soil particles. So if there's a massive rain or flood event, it doesn't wash away like an erosion. Mycelium has a very strong tensile strength. And now you're looking at a plant with and without. You know, which plant would you be? Or which plant would you want to be? So if you do not know anything about mycorrhizal fungi, and you are gardening or you're putting in a new landscape or a yard or house, start now. Put away the chemical fertilizer, because I'm telling you, that's what's killing the plants. They are become addicted to those synthetic fertilizers. So let's look at some of those mycorrhizal fungi that we all love. These are from South Carolina. These are black trumpets. Absolutely beautiful. They're out right now. People are finding them. Can't wait to get back. They're always finding stuff when I go out of town. These are my chanterelles from the low country, the coastal plain, South Carolina again. And these are mycorrhizal fungi. And there is no such thing as a bad mushroom, by the way. No such thing. If you think, oh, well, that's not good. It's just a turkey tail, or that's bad because it's a destroying angel. I disagree. They all do amazing things for our plants and for the environment. Yeah, you wouldn't want to eat it. What's that? Lobster mushroom. That's pretty interesting. That's a composite organism, big time. So that's a mycorrhizal mushroom that's normally all white, a big roostler or milky, that grows on the roots of a hemlock tree in my area. And it's typically not edible, but then another mold attacks it and completely changes the chemistry of this mushroom and makes it into an amazing edible, right? And it completely distorts the gills. So just a really cool mycoparasite. And then you have my morels, right? These beautiful morels. How many of you have a really good morel spot? Where is it? Do you want to share with us? No, somewhere here? Okay. That's what I think about mushroom hunters are the coolest, most loving people in the world, except for during morel season. You know, they carry knives. It's almost undebatable that all terrestrial ground orchids in North America require a fungus to open the seeds because the seeds do not contain enough energy to pry open the seed coat, right? It's like an oyster knife. It's like a little coil and it prongs in and it pops it open. Then it'll live with a fungus below ground, which feeds it. And, you know, they have a nice, you know, college roommate experience for a couple of years. And then finally, the orchid leaves come up and it starts to photosynthesize on its own. And then the orchid secretes a toxin and then kills its roommate, kills the fungal. It's like, what is that? Right? The fungus that does that is a root rot pathogen for a pine tree out east. So I find these moccasin orchids on mushroom hunts or whatever. I find them and someone says, oh my God, there's a moccasin orchid. And well, there must be a dead pine tree right behind me. And I look right behind me and they're like, how do you know? I was like, because that's the only thing that would work, right? I didn't even have to look. So we owe a lot to fungi, but here's how you can mess it up. And here's how we have been messing it up is synthetic fertilizers derived from natural gas sources. Mycorrhizal fungi, if they have phosphate in amounts of just like in the parts per billion, they will not associate with a mycorrhizal partner. Let that sink in. As we dump Miracle-Gro and Osmocote and crap on our plants, they will not allow that association to occur. So you need to let nature go. You need to let mushrooms bond with the plants and put the chemical fertilizers away. All right. You can inoculate your plants. It's not too late. It's like if you put these away, you could still inoculate mature plants at the interface environment, the root zone, the drip lines where your trees and plants are, right? Because that's where the mycorrhizae live. They don't live up underneath the tree, close to the tree. They're all on the root tips. That's it, right? But if you do this, you have sterile soil, and now you have a bacterial dominant soil, and that's bad for the health of the plants. So then what happens next? The fungi disappear, right? Now they have to eat your chemical fertilizer. And now they have no protection. So now what's next? Now you have to double down and say, oh, my plant has a fungal infection. Now someone has to, or a farmer has to apply what? Fungicides, right? So you see the domino effect. We can prevent all this by understanding mycorrhizae and applying mycorrhizae to our plants. This is a conventional cornfield, right? This is depressing, right? There's no soil production. There's no soil adhesion, right? If it's windy, the soil's gone. This is monoculture. These are clonal seeds. These are probably genetically modified seeds that have been sprayed, by the way, with something like atrazine, right? So there's all kinds of things going wrong here, right? We need to preserve our heirloom and ancient seeds, our genetics, and we need to plant biodiversity within the plant community, right? This monoculture is not going to work. It's done. So years ago, when I was at Clemson University, I received a fellowship for the EPA, and I had to go off to Athens, Georgia for a week. I was 36 years old. I had Mushroom Mountain, thriving company, and I was culturing some Petri plates. I noticed some contamination, and I was only there on the weekend, so I noticed some contamination on these plates. I put them in a Ziploc bag, and I threw them in the lab trash can. That's it. And then I left for a week, and then I came back, and then I noticed the fungus like surging over the top of another mold, and I'd never seen that before. Never seen it before. Years. I was like, that's interesting. So I took these pictures. I thought that was cool. And then I zoomed in. I noticed, I was like, well, the mycelium is usually a complete symmetrical circle. It's a beautiful, pure, symmetrical circle. But then it stopped and it surged over the top of this other mold colony, right? So it put its energy over the top. And then I drew some little arrows like this because my dad was in the military. And then I'm like, it looks like this fungus is attacking the mold. It looks like a surge, like an invasion. And then you can even see on this other slide, a slight depression right on the edge where the mycelium was pushing down. And then these little droplets at the bottom left. I know we have a lot of light in here. I'm going to pop it up like that. So then that's the same picture. So I zoomed in, checking the resolution, and I noticed all these droplets. And I'm like, that's weird. I've never seen droplets like that on this mycelium ever. Only when it's like attacking something. So I was like, I looked at it and then I was like, well, that's unusual. So it must be sweating some kind of novel antifungal compound to kill that aspergillus that was on the plate. Okay. That was just idea number one. And then I started looking at metabolites. So let's look at the sweat now. I was like, now mushrooms sweat. They make chemoattractants, they make enzymes, but now do they also make antimicrobials and disinfectants and all these other things? So what's protecting them from just, doing what they normally do? This is when people call mushrooms decomposers, I get offended because that's part of it, but they are composers. They create. They are like assembly lines. They have genetic tools to create new things. Yeah, they break down and use things. They need a food source, but they are creators. Don't forget that. Love it. Just love it. I love driving around the country. My God, I don't know if they still do this out here. In the middle of South Carolina, I saw somebody crop dusting a cotton field like just a couple years ago. I couldn't believe it. And there was houses everywhere, like farmhouses, families, dogs, chickens, just blasting it out. It doesn't stay in the field. It does not stay in the field. If these are pesticides, are they targeting one exact pest or two or three that are, they're trying to kill just a few insects, but what are they really wiping out, right? These are cocktails, often on the edges of, of water sources. And we do this with cattle too, with fecal coliforms. This is just a pesticide lecture, so but this does not stay. Now it goes surface and then subsurface. And so all these chemicals go into the waterways and they just move, right? They move very quickly. Bacteria are easy to trap in a mycofilter, by the way. That's a micron filter. Mushrooms are really good at trapping bacteria. Chemical degradation can take weeks to months. When someone says, oh, let's just mycoremediate that and put a little, coconut boom there and it'll filter out the chemical pesticides. It doesn't work that way. It needs time, right? It needs contact time for those enzymes to work. So where does that water go, right? Now we have pesticides in our water. Even if it goes to the water treatment plant, what are we drinking, right? What are we drinking? But it doesn't stop here. It flows down. I don't know how to. There it is. I had a waterfall up here. It just goes downstream. What goes up must come down, right? So this accumulates, and we have massive blooms of fertilizer blooms out into the Gulf, just killing everything, right? It doesn't have to be that way. Giving you two ways to reduce or eliminate the use of agricultural fertilizers is mycorrhizae. That's the solution. So this passes down in the food chain, right? This passes down the food chain. It hyperaccumulates. Things like silver nanoparticles now are big with the EPA. They're unregulated and they're antimicrobials. So anytime you see something that's antimicrobial, be suspect. If it's not labeled, it is silver nanoparticles. So what's another solution? If we have agriculture runoff is composed of fertilizers. Let's talk about getting rid of the pesticides, okay? I told you the bad news. We're going to do the good news now. So we have entomopathogenic fungi that are here, and they are everywhere. You can find them on nature walks. You can find them in the botanical garden. You can find them in your backyard. But where you're going to find them the most is where people don't spray, right? The fungi have set up a natural defense system there, and you can find them. So this is like a figure 8 life cycle. If you can see yeah, you can see my little cursor. So these molds can exist as a mold state or an anamorphic state, imperfect state, but along comes a spider or an ant or a grasshopper, right? What's interesting is those spores can sometimes be so host specific that they'll stick to the armor of that insect and they'll recognize the cuticle, the chemical composition of that insect. And then and only then will they drill a hole, a peg, and enter the insect to start to mummify and to get up into their nerve cells and around their brain and start to do behavior controls, which I love. And behavior modification, relocation, and even putting them in a position to do more damage, like a dirty bomb, to more of the insects that are involved. It's like weaponizing a little drone that goes back and finds more. These are zombie fungi, right? These are cordyceps. This picture was by Daniel Winkler. He took this picture in my backyard. Thank you so much. Daniel this is a week before he came to my house and to do a lecture. And I said, I think I found some cordyceps in my backyard, and we found like 100 or 150 in one holly tree. And I didn't see Daniel for like 2 hours. You know, Daniel's back there with his big ass camera, you know, and he was taking these pictures, but it also has a secondary infection on the fungus. See that on the tip? He thought that was really strange. So this is like multiple levels of biology going on. It's pretty wild. So this ant, you know, unfortunately got infected, and that's how they fruit upside down. You know, climbs up the limb, and most of the times it'll go into these. This is on the East Coast. That's a bud scar. It's very precise. So it finds a bud scar, it bites into it upside down, and then it melts the jaw muscles of the ant, and then kills the ant, and the mushroom pops out the back of its head, straight down, and it positions itself over the pheromone-laden highway of the rest of the ants. That's like a dirty bomb right off of the highway here to infect more cars. That's creepy, right? Everybody, feel the back of your neck right now. Right below, above your shoulder blades. Do you feel a little bump? Oh, Jesus. Was that there before you came tonight? Your job is to measure it. But I found so many of these. This is a spider. This is on a water oak in my backyard. Once I knew they were there, they were everywhere. Once I found the one, I was like, oh my gosh, I just need to look. I need to turn over the leaves or go outside with a flashlight at night. And I could see spots under the leaves. You know, it was exciting because they were just everywhere. God, thank you. This was my first cordyceps that I ever found. You'll never forget your first. And I was told to go to an old growth forest and look around. The trillium was like, 2 feet tall, jack in the pulpits. It was like it was beautiful on the East Coast. Nobody goes there. Like a little secret. And someone said, if you want to, a friend of mine, Rick Huffman, I'll give him props. Rick Huffman, founder of the Native Plant Society of South Carolina, said, you got to go to that spot because where there's rare plants, there's rare fungi, right? Obviously, I walk into the woods and in 5 minutes, I find these little threads sticking out of a log, and I pried it open and found a beautiful black carpenter ant, which I later turned into Harvard, and they said it was a queen, and they haven't seen that species since 1907. Just like that. That was my first. So I took a little dental pick and teased some of that mycelium out of her abdomen, and then I purified it, and then I caught more ants, black carpenter ants, and then I infected those. So I knew I had the pathogen because it's species to species. But then I started looking at the clades of ants, and I'm like, you know what? Camponotus, which is a carpenter ant, is very close to Solenopsis, which is a fire ant. Do you know what fire ants are? Do you have them here? I brought you some of those too. Kudzu and fire ant. You got the combo. I brought you some really, all females. So I decided to practice on fire ants. And I made fire ant farms in my house. This is a Friday night idea. This is like a 2-beer idea, right? Hey, I'm gonna make fire ant farms in the house. And my ex was like, no. I'm like, they're not gonna get out. All right. So anyway, I added the fungus from Camponotus to Solenopsis, and it mummified the red imported fire ants. We're talking about an organism that does $5 to $8 billion of damage in the United States alone in 1 year. It mummified them in 5 to 7 days. That's pretty hot. From a fungus I found in an old growth forest just sitting there on a log that nobody's seen since 1907. And there's more out there. There's more out there. And if you go to, well, I'm not going to name any names, but if you go to Home Depot or Lowe's or anywhere, and you see these big bags of what? Ant killer and grub killer for what? Yards and grass, cookie cutter yards and cookie cutter neighbor. It's like, it's so toxic. There's more pollution done by homeowners than any industry in the United States. Collectively, we can make the change. What if we all had a target specific fungus for Japanese beetles, emerald ash borer, longhorn beetles, pine beetles? Name one. All right, keep going down the list. Do y'all know that one? I hope I brought, no, I didn't bring these here. I'm not even joking about that. This is because they might actually come arrest me. This is the spotted lanternfly. This one's bad news. This is the #1 threat to agriculture in the United States, right? So it started in Pennsylvania, but it didn't stay there. It didn't take long. All right, its original host plant was the tree of heaven. And when it runs out of that, it eats everything else. And it loves vineyards, loves fruit trees, loves different ornamental trees. So once it runs out of things, it just covers them. That's what it looks like. It looks like a big ass leaf hopper, and it just sucks into the tree. The only remedy for this that they've found in Pennsylvania and other states, the only recommendations they've made was to spray and inject, and at the cost of $200 per tree. That is a lot of spraying. And if you think it only kills the spotted lanternfly, you're totally wrong. It kills a lot of different insects and other organisms, right? Other invertebrates. It's not target specific for that insect. Others are tree wrapping with adhesive tape, which traps woodpeckers and chipmunks and things. I don't want to show you those pictures. So I'm lecturing up in Pennsylvania. I show pictures of my ants and my beetles. And then a homeowner came to me one year and she goes, because I said, if you see anything strange, just like through the airport, if you see anything, report it. If you see anything unusual, report it. Now you know what an infected bug looks like, right? I'm going to show you more of those because I'm doing this again tonight. I'm showing you more because we need more bugs turned in. So she goes, she sent me these pictures. She goes, I think they're spotted lanternflies. Look at that. So she sends me these. And she found them in a little area in the woods in Burke County, Pennsylvania. I mean, ground zero. And she goes, I found 100 of these in a 10 by 10 foot area. And I haven't seen them anywhere else. So she drove them down to me from Pennsylvania, right? And that's what they look like. See how the fungus is coming out of the hinges of the legs and everything? That's not post-mortem. That was infected alive. And from that one specimen, I could just make millions of that, right? Highly infectious to that particular bug, right? And that's what's cool is we found a spotted lanternfly fungus that is more target specific to recognize a spotted lanternfly than anything else. All right, so that's strain. That's an ecotype kind of development. And this honestly should be free. If we learned how to take these, I'm going to teach a class online this year when the instars start to come out, where if we could take these, you could put them in a blender and blend them up in sugar, powdered sugar, and then sift the body parts. This is what I do on Saturday nights. What are you doing? Sifting body parts. And then you can make a spray, or you can gift some of that to your neighbor and then spray the insects on your tree, and they can take the next batch and just keep going like a bread culture. That's the only way I see it happening. Because I'll tell you right now, the University of Pennsylvania and Cornell found where my spot was, and they collected the same things. And now they're sitting on the same cultures, and nothing's been done in 4 years. Yes, I hope you're listening. That technology is here. There's already entomopathogens listed through the EPA, right? The same fungus, it's Bavaria, the same one is sold under a brand name, but it's not that target specific. That's called Mycotrol or Botanigard. It's better than the alternatives, which are chemical pesticides, but I'm telling you, it's still, things can be better. We found something target specific for a spotted lanternfly, and it's not being used. It's not that odd that it isn't. In my mushroom farm, I had everyone who grows mushrooms has a fungus gnat problem. And also with the cherry industry out in Oregon, they contacted me and they said, we really need to find a fungus to combat the spotted winged Drosophila. If you ever heard of those, those are pretty bad. It is a fungus gnat that infects or bites and feeds on the fruit right before they're ready to pick or edible. It's just like terrible, like right at that moment, right? And so I'm like, well, I don't know, but maybe I could buy some fruit flies and then try to train a fungus to grow on them. But then something even more cool happened as I was lecturing up at Penn State, and another lecturer came on talking about chemoattractants of flies or octanol that attracts insects, like I talked about. And they said mango yeast, if you want to attract fruit flies and trap them, if you take mango, mix it up in a little bit of soapy water, you will attract all the fruit flies. That's really high. That's a yeast producing octanol, very high. I thought that was cool. But what he said was one female virgin fruit fly one female, dead or alive, would attract all the males in a space that was like 10 by 10. One dead female fruit fly, that tiny, if you put it on a sticky mat, would attract all the males. They would stick to the mat and they'd be done. That was it. So I thought, man, that was pretty cool. And then so it's Christmas, and I'm like thinking, people are asking me, what do you want for Christmas? So my ex was like, what do you want for Christmas? And I said, I don't know. She goes, well, just get something for yourself and I'll wrap it up. So I bought 100 female virgin fruit flies. All right. Because I'm a minimalist. And so I bought them, and then, they came, and then she came, and then she was like, can you explain something to me on this credit card statement? I'm like, what? Because it said just description. It said live 100 virgins, ... I'm like, it's Christmas. They were $1 each, so it kind of made sense. Anyway, so I infected these female virgin fruit flies. That was my Christmas present. And 10% of them like colonized, the rest of them didn't. But then you take those 10 and then you plate them out, and then you add those to another 100, and then you get like 50%, 80%. So you're training that fungus forward for cuticle recognition. That's how you classically train these fungi, right? It can be done. Oh, and I wanted to really infect all these females, at least have their spores in their body to release into the fruiting room, which is like weaponizing the females. And I gave that lecture at a university, and there was this freshman male in the front. And as soon as I said virgin, he woke up.
Huh?
And when I said weaponizing the females, he was like, well, what do you mean? I don't get it. I was like, imagine going to the club and every girl has Ebola, but you don't know it. He goes, dude, that's cool. I was like, that's the dark side of the force. Weaponizing the female virgin fruit flies. That's what I do on a Saturday night. And there's destroying angels. I told you, like, not all mushrooms are good for us, right? Ingest it. They could be good medicinally. There's something about every mushroom, right? Something about them. There could be some genes in there, right? Programmable cell death, cancer cells, maybe some kind of delivery system. Man, you don't want to eat one of those, do you? This is our Eastern variety. And I get a lot of calls. I'm on the toxicology for NAMA. I get a lot of calls, and I get this call from Florida, and it's on Sunday night, and my phone is ringing over and over and over again. I do not recognize the number, so I don't pick up. And finally, it rings like 10 times, and I pick it up, and I'm like, thank God you picked up. My dog is sick. It ate a mushroom. I'm like, can you describe it to me? It was that. All right. Chewed up and white. And I'm like, what kind of dog do you have? And he was like, I don't know what breed that is, but he was like, well, it's like a 1.5 pound baby Chihuahua. And I'm like, what? And it's already has cramping and diarrhea, and it's in a corner sitting down vomiting. I'm like, ugh, this is not good. So I told him what to do. I told him to get Legalon, go to the vet. I talked to the vet, and usually with diarrhea onset, that's liver failure, and that's at the point of no return. So I waited about a week until I gave him a toxicology report, a follow-up. But before I did that report, I opened up my mailbox at the farm. I opened it up, and there was a little card with this picture on it. And it just said, thank you. You saved my life. Garbanzo. Garbanzo, the ammonite eating dog. And then, that's what choked me up every time. I started crying at the mailbox, when I saw that, the sweater. And then I was lecturing in Florida like a year later, and I'm lecturing like this, and there's a guy standing at the back, and he's holding a dog in a sweater. I'm like, that can't be him. And then I walk out after, and he goes, Garbanzo wants a selfie. Listen, I picked this is a psilocybe mushroom right next to the destroying angel in Florida. That's a really cool picture. Um, one's deadly. There's no cross contamination here. That psilocybe is pretty fine to eat by itself. The good news is if you don't know what you're doing, no matter which one you eat, you are guaranteed to see a light. And also years ago, so I was interested in deadly mushrooms. I'm like, oh, well, the Meisner test, if you squeeze a deadly mushroom juice onto newspaper and water as a control, that's the control on the left, and then you hit them with hydrochloric acid, concentrated hydrochloric acid. If it turns blue, that might not be good. I mean, it's not 100% accurate, but it's pretty good. So this was a Shiloh was the dog. It's a famous Meisner test for this dog. So I was like, well, how can I make this more accessible to people? Why can't we have like a little test kit, right? That we take out into the field and we squeeze the mushroom on and then it just turns a color. And I'm thinking, Jesus, something like that already exists. I was like, it's like oh shit, it's in the picture here. It's a pregnancy test kit. Right? It's a pregnancy test kit. And I'm like, okay, I'll just open it up. I don't have to 3D print anything. I'll just take out that paper, replace it with this. I'll just remodify the capsule, right? But then I'm like, went to buy some just for, R&D. And I'm like, man, these are expensive and I need a bunch of them. So I went to a thrift store in South Carolina, a very conservative Christian thrift store where they sell expired stuff. And I found a whole wall of expired pregnancy test kits. So I went in there, I was like, oh my God. So I got my shopping cart and I went over there and I just like raked them off the aisle, like brrr, they went in. I had a whole pile of pregnancy test kits. And I came up there like smiling up to this lady and but she like held her neck, she was covering her cross, like, oh my God. She goes, would that be all? I was like, oh, you got some more in the back? And nothing beats my call with the Amanita muscaria, the guy from Virginia. Listen, these are not for ingestion. I don't think there's psilocybes are way more accurate, way more easy. But someone called me from Virginia and said, hey, thank God you picked up. It was like 1 in on vacation. I just ingested Amanita muscaria and I vomited. There's shadows moving. Everything's breathing. I'm like, that sounds pretty normal. And I was like, sounds like you got through the worst part of it. And he goes, no, man. He goes, the worst part is my cat won't stop staring at me. And I'm like, okay. I'm like, what kind of cat do you got? I said, you know what? That's what cats do. They just stare at you. And then he goes, no, man, you don't get it. And that's when I knew it was about to get good. Right? He goes, when I lock eyes with my cat, my head won't turn. It's like a tractor beam. And he said, there's zeros and ones flowing like back and this like information. And he said, I swear I could feel that this cat is brainwashing me. And I honestly said, that's what cats do. I was like, just put him in the other room, drink some water, turn off Fox News and go to bed. Play some music, rehydrate, just chill, be good. And he goes, I tried it. He goes, I put the cat in the bathroom, but there's a little gap under the door. And then his little leg comes out. It's like freaking me out. So he kept freaking out. Anyway, he was like, how long is this going to last? And I'm like, waited about 5 seconds. And I'm like, forever. All right, last fun one. We'll get it back to business. This is the intermission. Oh, I got a call from this lady, and she's like, this is the South. Love doing the Southern talk. She goes, can you identify a mushroom for me? I was on the phone. I'm like, yeah. She goes, I don't know how to tell you this. but it looks like somebody's done buried a dog upside down in the mulch. And I was like, I know exactly what you got. That's the dog penis stinkhorn. And she was like, oh my. Is it edible? Real, that's a real phone call. I don't get paid for that. This is my bonus. These phone calls are great. Anyway, let's get back to it. So listen, fungi and plants have evolved. This is cool. These are some hot thermal vents around Yellowstone. And you know what's interesting is where did those colors come from? Does anybody know? Bacteria, yeah, thermophilic bacteria. And right on the edge over here are actually plants. And this soil and the water over here, the subsurface right at the water level is about 155 degrees Fahrenheit. Now tell me, can you plant corn, tomato, or a squash in 155-degree water? Will it survive? So how is that possible, right? How is that possible? This has been known for some years. So there's a plant fungus virus, three-way symbiosis at these pools. Look at the grasses right here. They're right on the edge, right? Their root systems are in blazing hot water. And so they found a fungus living inside of the plants. That's an endophyte, right? They live in the roots, shoots, and leaves. They found that, so they figured that was it. So they took the fungus, they purified it, and they put it back in new plants that didn't have it. They put them in the water, and the plants died, dead. They're like, that doesn't make any sense. That's the exact same plant species. What happened? And so then a virologist looked deeper, and they found a virus living inside of the fungus that was living inside of the plant, a beneficial heat-tolerant virus. Interesting. Something we might want to look into, right? As the world heats up. Or for desertification, you know, in areas that are really dry and hot. So that was the non-colonized, that was the curvularia, the fungus, and there was the virus inside of the plant. I was in microbiology, the journal. So endophytes, this is a big thing. Yeah, I've been studying mushrooms for a long time, but every plant, every flower, and I just happened to be in a botanical garden. If you go out there, can you imagine the level of mycodiversity that is here with the plant, the botanical diversity here? It would be crazy. Like if you punch every single leaf, you will get something different, even on the same plant or even a petal. Because what these plants do is they carry the fungal genes forward. Like they pack them into their little time capsule, their DNA. So when you're planting a seed, you are planting a composite organism. If you're not buying a genetically modified seed that's been treated with something. That's why saving seed is so important. You know, there's a reason we do that. Our ancestors did it. So we save those heirloom seeds of the plants. Why would you save seed from a plant that got downy mildew or, you know, verticillium wilt or, you know, spot disease or, you know, root rot? Why would you do that? That plant it didn't have what it took, I guess, right? But the one right next to it, who was exposed to all the same pathogens, is completely fine and green. There's a reason. It has endophytes in it. It's got natural fungal filaments that are in its leaves trying to protect it, right? And these are invisible. So the fungus threads its way through the seed, up through the plant, through the leaves and the shoots, and then it deposits itself into the seed for the next generation. How about that, right? If you take cuttings too soon, you're not going to get the fungal endophyte, right? If you take green tips, that's why second year wood is actually better. So I actually taken endophytes from one plant and stuck them in the other. It was actually a trichoderma, which is a mycoparasite. It's a mold. I grew the mold out on toothpicks or matchsticks, I think, And then we drilled holes in some grapes and muscadines that were having a Botchysferia outbreak or a leaf spot fungal outbreak, where they had to spray systemic fungicides in the vineyard. And we drilled and put in these little matchsticks of the fungus, and it caused a phytochemical response, and it completely eliminated the Botchysferia in 1 year, the following year. We are a composite organism. Hope most of us know that, right? I am not trad standing up here. I'm like a coral reef. Trad's like the shell, and there's a lot more going on in here and on there. You know, we're outnumbered. Our gut flora contain bacteria, they contain fungi, they contain all kinds of organisms. That's why we are not what we eat. We are what our bacteria eat, or our microbes mostly eat. Got to be very careful with what we put in and on our bodies and protect it. Back to the metabolites. This is, you know, I was very interested in that little battle we had going on between the mold and the fungus. And so I started to intentionally plate things out, fungus against a fungus, just to see what they did. So that on top is a shiitake fungus, you know that one. The one on the bottom, well, I guess I could say this in Colorado. That one is a psilocybe species. See, in South Carolina, I'd say it's a oyster mushroom. That's a psilocybe species. So look at the droplets that it's creating. You have a psilocybe that grows in a very complex environment, right? Dung ridge, lots of bacteria, right? Faster growing, shiitake is very slow growing. But look at those line of droplets that it makes, those antifungal compounds, just absolutely beautiful. They are dialing up antifungal compounds specifically to kill shiitake. They may kill other fungi, but they're going to definitely kill that shiitake fungus if it touches it. All right, so now I take plates, do some bacterial plating. Got a class 2 lab. And then so Christmas, I got my virgin fruit flies. My birthday is in January, so it wasn't long before I needed something else. And again, I was asked, what do you want for your birthday? And I was like, I guess I'll just get it and you can wrap it up. So I ordered salmonella. It gets better. So I ordered salmonella, E. coli, staph, streptococcus pneumoniae, streptococcus pyogenes. These are level 2s, by the way. These are infectious. And Pseudomonas and Candida. So I ordered like 7. It was like the combo plate with the FDA. I'm probably on a government watch list, by the way, especially for the virgin fruit flies. But I ordered the combo plate, and the reason I got those, it just wasn't random. It's because all of those exhibit different cell wall properties, right? And also different motility properties. So I wanted to see what these fungi are capable of doing if I put them in the same gladiator match that I did with the fungi. So then I started culturing. This is actually a staph culture off my body. Just I was curious. If you ever need, if you are doing cultures, if you're ever curious about what's growing on your body, don't. Do you know what's living inside of your belly button? Mm-hmm. No, you don't. No. We'll be doing a body shot later just to kill it. There would be no more body shots if you knew that. Yeah, no. Anyway, it can take as many as 12 years of development. That's probably sped up with some time and AI and things like that to find and isolate antibiotics from plants, from bacteria, from fungi. There's so many sources of antibiotics, right? But fungi are right there. They're kind of sentient. It's like when they react to something in their environment and they're able to create something on demand, that's the rabbit I was chasing down the rabbit hole. I was like, that's something, nobody's done that before. But, 12 years and then 2.5 years for bacteria to become microbial resistant because bacteria are promiscuous. Does anybody know what that is? All right. It's like the nightclub where, but bacteria of different species, they can link up and they can do gene transfer of drug resistant genes, and they can just adopt those and give them away. Like, hey, you want to come to the party? Here's the keys. Swim over into the antibiotic gradient and exist and just start multiplying. That's scary. That's what's going on. The overuse of antibiotics in agriculture when people get the flu and they take antibiotics, it's a virus, for God's sake, just take antibiotics for everything. Feed it to the chickens, feed it to the dog, feed it to the cats, feed it to the cows. It's in our drinking water. Shocking. Love city water, right? Full of stuff, We need those extra vitamins, So I created these arenas, fungi, bacteria. These are little pellets that go on plates. That's what they look like. So this was my birthday present to myself because I really saw this as like, this is my present, is just knowledge, like learning from fungi, learning from them. And then I would set these pellets up. This is E. coli and shiitake, by the way. Shiitake didn't do that good, but it did okay with the E. coli. It didn't do so good with everything else. Shiitake is a slow-growing fungus, very limited in its food source. If you really want something that just absolutely has, the skeleton key, you go after like oyster mushrooms and things like that. Polypores are really good. So it sat there for a couple of days, and then all of a sudden you see the bacteria just windswept over here, and you see this windswept too. So it's diffusing like low molecular weight antimicrobials into the agar. All these white cells are dead, but look at the way it went to the farthest part of the plate, like there was a tiger in the room. All right, and rep after rep. So then I started saying, okay, well, let's look at some research someone did on polypores. And this was Fomes fomentarius on E. coli. I had E. coli. Why not try it? So then remember the little droplets on the plate, those tiny, tiny little droplets. I'm like, that's really cool and all, but I need more. So if I had a little plate, what if I had made a big block of mycelium? What would it do? How would it react, to a little bit of E. coli? So I pushed down, made these little swimming pools. This was my intention was, my hopes are to take this to the throat culture stage, something topical. But the ability to take a culture like this, inject it into a bag. You see how it created a hydrophobic well, which was really convenient. I knew that was going to happen with a polypore. It would not happen with oysters or anything else. That culture would go right through the bag. So tensile strength was really important when choosing the species for these trials. So I had to have a mushroom that created this really hydrophobic well, which are the polypores mostly. And it sat there, and in 24 hours, there was controls, and there were E. coli dosed. So the ones on the left are E. coli dosed. It produced that fluid and that browning reaction within 24 hours. And then all of the control bags did not produce any metabolite whatsoever. Shocking. Not really. It was defending itself, right? It was producing a novel substance and quite a bit of it, coating its body with it. So much that I was able to pull out. You see there, Fomes fomentarius, E. coli trigger. And I was able to pull that out. Now, this is not sterile. Who knows what's in it? There could be some, bacteria involved. So I take this and then I use a syringe filter and filter it out. But I did this with maitake. I did this with a lot of different species, turkey tail, pink oyster, elm oyster, candidates that I thought would be good because I did the Petri plate study first. I saw what they were doing in a behavioral gallery, the little gladiator matches first. Then I would make the larger prototypes. So that's filter syringe. That was prototype number 2, like the upgrade. I'm like, okay, I don't want to make these wells. I'll just make these little indentions, these ports, injectable ports, like I can rehydrate them. And then I can inject there and then pull out the metabolite here on the bottom. That seemed pretty cool. So, and what's interesting about this is it just wasn't efficient against these pathogens that I ordered. It was efficient against fungal pathogens for agricultural pathogens. You know, botrytis, for strawberries, some major ones that, you know, all our strawberries are oversprayed because of these fungal pathogens. So we tried that, inject it there, out comes the metabolite. It's on a little riser, but then that kind of got a little bit messy. What was interesting is that 20 milliliters that I sucked out of that E. coli, when we diluted it 10 to the 6, was actually just as potent as the original. So that 20 milliliters can make 528 gallons of spray. So if that strawberry farm had a little outbreak, they could take a very small sample, inject it into a bag, get maybe 20 milliliters, put it in a 528 gallons of spray and treat their field within 48 hours. That's how fast it is. And more target specific. So shiitake staph did not do the best. The best for staph was chicken of the woods, orange mock oyster, and jack-o'-lantern. Unusually, all three are orange, and all three mine sulfur and smells like sulfur. So staph has an Achilles heel of the sulfur atom being wrapped around other atoms. And that's where most of our staph drugs are based out of. But the chicken of the woods created a new sulfur-based molecule that was target specific, not just for staph, but for MRSA, for drug-resistant staph over at Clemson. That's crazy. It's a huge opportunity for drug discovery. Turkey tail did really good on salmonella. I've never seen anything like that compare. So turkey tail and salmonella. I make these cutting boards, you know, at home. We already smear the mycelium. It's in my book. And you can smear mycelium on the cutting board, let it colonize and scrape it off. And you have a living cutting board with turkey tail in it. No problem. Naturally antibiotic, living cutting board. Reishi did the best against strep throat. So we took some of this fluid, we mixed it with honey, we hardened it into a candy, we made little lollipops, we made little streptopops. Not on the market yet. So we test these with inhibition studies, MICs, minimum inhibition concentration. You look for little halos around. So I have little discs of antibiotics that are already on the market, and we're looking at different inhibition concentrations comparative to what's on the market, if not better. But more importantly, could they be more target specific to the different bacterias, right? The gram negatives and the positives versus both. And that will lessen our, you know, that will lessen the drug resistance problem, right? So there's clindamycin on the left, and that's completely drug resistant, and this one is not. So that means it's working. Doesn't do anything to that bacteria. You don't see it working at all. Yeah, that's chicken of the woods. It performed the highest. Jack-o'-lantern actually might have beat the chicken of the woods. I've seen if you put a jack-o'-lantern strain on the same plate as MRSA, I've seen it completely climb over, graze, and just carpet bomb the hell out of it with metabolites. It's very aggressive. So we tested a lot of different strains against a lot of different bacteria. And that's the dilution. Let's say, 10 to the 6 would be active. The slide was out of place. So recently, I presented this at Telluride last year. I went from the big bag to the medium bag, and now it's shrunk down to a tube, which is just as efficient because this is more patient specific. So what I'm looking for is a more target specific and patient specific, where you can come into a hospital, they do a throat culture, a robot injects it, and then you get your novel antibiotic or treatment just for you out the door within 24 to 48 hours based on your little biopsy. So those are the tubes. I've already got them made. I've already tried them out. And so the robot goes in. Right now I'm doing it by hand, but it's basically set up for the same machine. It's pierced through the injection port. They have little micron filter patches, And the pathogen goes in. It's not blood. It's just red to show you what it is. It also has a selective and amplification solution in it. So that actually feeds the bacteria to get them to multiply really quickly. You know, you're giving the bacteria what they want too, right? So you amplify it. Now there's a lot of cells there. The fungus reacts. You fill that, you rehydrate it through the bottom port. Took me a while to figure that out. And then what happens is the novel metabolite sweats off of this cone because there's a space, and it goes into the reservoir of water below, and then you can extract it here. So there's no contact between the pathogen and the novel metabolite. It was much safer. So I'm looking at different machines to do this. And my goal is to get this under wraps like this year. With the patents, I've got a device and a process patent in order to protect it. It's way too important because what I see is a patent that basically, if I license this to someone, then they could invent or they could patent that molecule. So it's a patent that creates patents, which is really, we got to get a handle on that one. So I do not want anybody to shelve any part of it, so I'm trying to protect it with some really rich hippies. And they know who they are. But yeah, I mean, could it work against viruses? I don't know. It's like, and some folks down in the CDC when COVID hit, you know, they wanted to try my method, but they wouldn't even let me because they said the vaccine people were already locked in with their contracts. So we couldn't even do any R&D with it just to try it, right? So I'm excited about it. Excited about moving forward with that. Not so excited about what we've done to the planet. You know, what mushrooms can do for us. They are making bioplastics now out of mycelium, right? And hemp and different plant waste that decomposes and not like, hey, you have to put this plastic bottle in a commercial composting site, and it still takes like 100 years instead of 1,000. These are things that can just melt in the ocean, right? Melt. You know, we need plastics. We need things that are pre-inoculated with fungi, right? If you want to know where the fungi are, you know, build it and they will come. We should be going out into the ocean. We should be going to the landfills and we should be digging it out because they're there. They showed up. We made the mess and they're there to try to figure it out, and we can culture it off of them. But, you know, the good news is, you know, there are some fungus that have been found, and there's been other species. This is old news. Pesticides that are fungus, polyurethane degrading. I remember when this came out years ago, they're, oh, mushrooms are going to eat all the plastic. That's not right. They can eat pure polyurethane in the lab, but that's, you know, that's about its limitations. They're not going to break down a big pile of water bottles out in the ocean, right? What's exciting is there is something out there, you know. I'm an optimist. Right. There's motor oil. They can eat motor oil. Mushrooms are adaptive. They lived in sand dunes. These are found off Kitty Hawk, like North Carolina, like on the dunes, right on the ocean. If you don't think that there's mushrooms out there, on the beach, in the lapping surf, think again. Right? They are. They're associated with dune grasses, and they're receiving a lot of salt, which is kind of rare. And if those fungi can degrade hydrocarbons and they can be a part of the degradation of oil spills, there's your solution right there. Because that was the limitation of the BP Horizon oil spill, was there was not salt-tolerant fungi available to use. You know, herbicides, atrazine. I've trained everything on all these different pesticides. At day 10, this is 3 times the labeled rate. Fungus, figure it out. There's nothing on that plate but atrazine, right? This is a red flagged pesticide by the EPA. Well, what used to be the EPA, right? When I was there. And these fungi figure it out very quickly, and they can break it down. We did a study with Furman University. I'm not sure if it's published yet, with atrazine. And now you probably wonder, well, if it breaks down atrazine, what happens to the atrazine? Does it go into the mushroom? Does it go into the soil? You know? So they did this study and they found no detectable atrazine in the fruiting bodies, which was going to be a very well-cited paper, by the way. I'm like, let's go after low-hanging fruit because everybody wants to know what happens to those molecules, right? And of course, we don't want to, we don't want biotransformation. We want bioremediation, which takes fungus and bacteria and everything working together dynamically, right? Fungus can biotransform to a certain point, but it needs partners. It needs a little septic tank to work that out. It needs a relay race of different microorganisms. Biotransformation could mean, hey, well, we don't see atrazine in there anymore, so it must be gone. Well, it could have melted down a sword and made a gun. It could have made something more dangerous. Does that make sense? We want bioremediation. So I'm going to finish with our favorite subject. Cheer everybody up. Psilocybe cubensis, Schedule 1, right? Still Schedule 1 federally. No medical use whatsoever. And highly addictive. How many people do you know are addicted to mushroom? Oh, this is a bad crowd to ask. The wrong state. I mean, maybe I was when I was young. I don't know. But no, it's almost developing an aversion to it. Yeah. I mean, I had experiences a lot when I was younger, and it's like less and less every year. And that's called permanence, when you reach that state. And, I use it calibration once a year, maybe not even once a year, maybe after a life event, right? Maybe after trauma. Anybody ever had trauma? No? Okay. Yeah, I hear that on retreats a lot until they take the mushrooms. Right. We've locked so many things away, but it's just, Schedule 1 federally doesn't make sense. And I was just saying, when I see this slide with my daughter, it's like unconditional love, right? Should be legal, right? That's chicken of the woods, by the way. Unconditional love. I'm telling you, our kids are watching us and they're learning from us, right? Like my daughter's learning from me. There is nothing wrong with it, right? Nothing wrong with not taking pharmaceuticals that cover up. If you can't feel yourself, you can't heal yourself. That's how I feel about it, right? How many deaths have been associated with pharmaceuticals in the last year? Well, let's do pre-COVID. That's an interesting statistic. Pre-COVID, 30,000. COVID, 130,000. Jesus, right? And they say, well, give me one reason why, this is better than pharmaceuticals. And I could say, this is from Robert Rogers. He's probably listening. Thank you, Robert, for this line. He goes, I could think of 130,000 reasons why pharmaceuticals are not good, and not one against psilocybin therapy, right? In a safe and productive way. That's what the state has done. So thank you for what you've done in the state, right? Let me tell you about my first trip, and then we'll get moving on some other stuff. How many of you have never tried mushrooms before? Put on my prescription glasses. You see, I like that. See, if I would have done it the other way. How many of you have ever tried mushrooms? God, you guys are just so honest. I love you guys. I can't do that in South Carolina because people are afraid to raise their hand. I've got a job. I'm a doctor. I'm an attorney. I work for the state. They ain't going to do this. Those of you who raised your hand, where are you? Right there. You just want some. I know you've tried them before. I've got 5 grams taped under your chair to take. I knew you were going to sit there. So my first time, 20 years old, I knew about him a little bit, didn't know much about him. And then I was in a band. My drummer, I was 18. Drummer was 25, much older. And we picked a bunch of mushrooms, but I didn't take them. I didn't know what they were. And one day we were watching the presidential debate in 1992. He was watching the debate. I was not into politics. I was 18. And he's watching the debate. And he goes, you should really try some of those we picked. I was like, what? All right. So I go in there and I made ramen noodles. That's all I had. I was broke. and made ramen noodles. And I just cut up like 10 of them fresh and just chopped them up and stuck them. Water turned blue. Noodles turned blue. I did not dose accurately, by the way. So I ate the whole thing because I was hungry. And then I go back and I sit on the couch and my drummer's watching this, right, on the big screen TV. And I start to just like look at these people, especially Ross Perot. They're really short, little hobbity looking dude, right? And then I'm like, start looking at him like really closely with high resolution. I'm like, oh my God, it's kind of weird looking. Big ears sticking out like this. And then I just started feeling a little tingly. And then my drummer on the wall had a steer head and a zebra skin. It's very barbaric. I didn't like it. It's very alpha male. So I'm looking at this. Then I'm looking at the screen. I see like this. And then I look up at the wall. And I look at him. And I look at the wall. And then I looked at the screen and I see that. I'm not kidding you. I was like, "Ah!" And the drummer goes, "You're ready." Put me in the control box at the studio. We had a recording studio and I sat on the ground and I listened to like the Double Doors live album for like 2 hours. It seemed like about 10 hours. But then I started to see things. I really went deep. Saw patterns, fractals, but then you just start going really deep, like really down the rabbit hole, down the black hole, down to that singularity moment where you feel compression, a little anxiety and fear, and then you can just release back into that white hole and invite everything back in, right? You create a vacuum of goodness, releasing that. It's such an amazing experience. I think that changed my life forever. And I got a job at a mushroom farm, 2 years later? That was it. I quit my job, just like Gary Linkoff said. I called the bartender and I said, I ain't coming into work today because something profound has happened. And that's it. That was it. We do perform retreats here. Colorado, we're getting certified now. Costa Rica and Jamaica. And that one was in Costa Rica. I had consent from everybody for that video. That's Irene in the background. And we do this kind of a mixture of traditional and modern. Blindfolded, music in the room. Some have headphones. Therapists and nurses on staff, everything. So I love what I do. That's Irene. She's the Blue Portal. She's the lead therapist. We do have a few more. Gia has just joined our team. And we also have Cotter, she does more of the traditional cultural shamanic methods as well. Listen, mushrooms are trying to teach us something. So this is the Rosetta Stone. What I've taught you is I've just kind of paid attention. And I know a lot of you have too. If you've really paid attention, mushrooms will teach us how to survive. We know that we aren't in a good spot. But again, I'm an optimist. I'm not a pessimist, but I'm pretty sure what we're doing right now, I'm pretty optimistic that shit ain't gonna work. All right. We need to read the language. We need to follow their lead. Right. We need to learn survival training. They've been through this before. Right. We need to learn. We need to be a fungal-based society if we are to understand and survive the next millennia. We can't solve our problems with the same thinking we used when we created them. Right. Storms are getting stronger. I live not too far from Asheville. Did you see what that storm did to Asheville? I've never seen anything like it. I've lived all over the place in the Middle East. I've seen towns leveled by bombs. Never seen towns completely wiped off the map. Right. So I went up there and taught mushroom production, wood chips. Scott and I with the trade school, we went up there and taught people how to mill wood and, build structures, and on the spare wood, grow food, filter the water. It's like all that stuff. Storms are getting stronger. Fires are getting hotter. Land's getting drier. We need soil creation. We need to hold out water where it is, right? So I think this mushroom knowledge, we need to really understand that it could take 500 to 600 years to make 1 inch of soil. And in my area, we've lost 15 feet of topsoil in the last 100 years. It would take 79,000 years to put it back. And we lost it in 100 years, right? Mushrooms can make 1 inch of soil in 12 months if you know how to do it, right? We just have to build it and they will come. Put the wood chips down. These forests, right? Look at what shows up first. They're the first responders. Cut a tree down, see what shows up. Those endophytes are already there, by the way. They're in the wood, right? They're just waiting for that tree to die. So the next time you're walking through the woods, like maybe a few grams, feeling it, right? Really feel the vibration of the forest. Really feel what it needs. It's going to talk to you. Hopefully it says thank you for everything you've done, because that's what it's told me. But it also says to me, I need help. I need your help. I'll help you if you help me. Okay? So let's help the forest. This is the kind of forest I want my daughter to grow up in. All right, that's Heidi. I think Craig's not Heidi, right? She's cute. Now she's 11. So this is her working in the lab. And she was looking at culturing plates. I don't know what that is. Probably legal, I'm sure. In her little tutu glove fit, you know, and then that's her getting older. Right now, she is like a little myco warrior. You know, they don't teach fungi in school. They say soil is non-living up till grade 5. That's the national teaching standards. So, you're laughing, but you're kind of laughing in disgust, right? What is going on? Soil is non-living. Wow. I guarantee you Heidi doesn't. That's not the truth. Did you know that she brought me home the paper? She's in 5th grade now. Last year, she brought me home a paper where she got a 90. She's a straight A student. She got a 90. I'm like, which one did you miss? She goes, oh, I said soil was living. And they marked it wrong. Holy shit, I about went freaking nuts. I still have it. I still have to go talk to the whole school department. I mean, like, what is that? Right. Well, it's public school, you know. But there's Heidi, and I'm just watching her develop. Look at her. Loving it in the myco lab. You know, our next generation. That's why I said the children are watching. We got to teach. If you know anything about fungi, if you know how to grow a little mushroom culture, teach in school, volunteer at an elementary school, and let them take a little thing home, you know, and teach them about recycling. Teach them to get the cardboard container, not the styrofoam or the plastic one. You know, teach them consumer responsibility now, right? We got to teach them. Everybody needs to know survival training. So this is what I do on Friday nights too. I cut out little shapes because I like to watch peace grow. It's like Shroom TV. It's free. So if you have peace, you have love. If you have love, you have happiness. So that's it for tonight. That's my book. You can buy it online, or somebody may have it here locally. Organic Mushroom Farming, Mycoremediation. I am working on a new one as we speak. So I'm working on this weekend. It's a very difficult book because this was a appetizer for the next one. And so the next one is going to be all these fungal solutions for all these different industries and agriculture and medicine, veterinary medicine, which is really important, and then different industries. That'd be the one I really wanted to write, but I had to write this one first. That's where I work right now with a good friend of mine. He does resilience training. If you're interested in that work, that's the guy, Scott Bunn, man. He's amazing. He's an engineer and he's all about resilience and survival and really an amazing entity, I would call him in a good way. I don't like the word entity, but spirit. So that's Treehouse Trade School. There's a thing you can look at that might, if you want to go to tradcotter.com, you can go to that. But anyway, give me some time to update it. I'm really busy with my book and traveling. And so if you just go to trycotter.com, you'll see that. And that is it. That's all we got. Well, it's not all we got. I don't know what time it is. Like, I could probably went way over. What time is it? All right. Thank you for coming. If you have to sneak out, don't forget the grams under your chairs. See everybody checking every chair on the